All right, so my name is Gerald Haynes.
Right, my name is Lily Dover.
I'm Nicole Webster.
I'm Amy Rogers.
I'm Ava Restrepo.
This is our cultivating prokaryotes in the laboratory and methods to detect and measure bacterial growth.
Right here we have the principles of growth, and we are talking about how bacterial and microbial growth is measured by an increase in total number of cells, not regarding their size. The cell number increases by a cell division process called binary fission for prokaryotic cells. The image to the right, so that right here, shows us how that works. Because the cell number is doubling in population each time the cell divides, this can be described as exponential growth, also defined as generation time. Clostridium perfringens is a rapid growing bacteria, and it has a fast generation time of about 10 minutes, which is crazy. That means in just 1 hour, 10 bacteria can turn into 640 bacteria. And generation time varies depending on species and other factors like environmental conditions. You can calculate bacterial growth with generation time using the equation NT equals N0 times 2N. And this little image to the right kind of shows you how that works. And NT represents the number of cells at a given time. N0 represents the initial number of cells you are starting with. And little n is the number of generations. One generation is the time it takes for the population to double.
Okay, so I'm going to be talking about measuring growth, and there are many ways, but I'm just going to be talking about 3 different ways. So the first one is the direct cell count, and this provides a really good estimate of total cells, but can't really distinguish if they are alive or dead. So it's kind of the only downfall with this one. But a liquid is added to the hemocytometer, and this allows for clear visibility. It's a special glass that is added to be able to count all the cells. And the slide contains a thin chamber that holds the volume of liquid, allowing for a very precise number. Another downfall is you need a minimum of 10 million bacteria per milliliter that is needed. And then another one is the Coulter counter, which is the one on the right, this picture. It's just an electronic instrument that is used to count cells through suspension. The liquid contains electronically conducted fluid as the machine detects the brief changes from the resistance that happens from the non-conducting particles as the bacteria passes through. And then the way they find the total amount of cells is just as they pass through the narrow channel. And then the flow cytometer is pretty similar to the one that I just explained, but it uses light scattered by the cells and uses a laser instead. And this can be used to count all the cells or just like subsets, and the subsets can be tracked by using tags or dyes.
So here we have our growth curve. There's 5 phases. The first phase is our lag phase. This is where our enzymes are synthesized required for growth, so the cells aren't growing quite yet. The cells are metabolically active and the cell number does not increase. The second one is the exponential log phase, where the bacterial cells begin rapidly dividing. During this phase, the cells are extremely susceptible to antibiotics. We also have amino acids and ethanol produced and the formation of endospores and capsules. Next is our stationary phase, where the total number of cells remains constant, but dead cells will burst, releasing their nutrients, but other cells will continue to grow and become more resistant to drugs. And our death phase is when the number of viable cells starts to decline, and then our phase of prolonged decline, the cells continue to die, but the fraction that does survive will adapt to the worsened conditions.
So I will be going over microbial cultures, and to begin, we'll start with culture mediums, which have a process that consists of nutrients dissolved in water via liquid broths or solid gels, and they can be refrigerated on an agar slant, and they're usually frozen in glycerol solution. Pure culture involves the replication of a mother cell forming a colony, and this was a method developed by Robert Koch, and this usually focuses on a single species. A stock culture is maintained continuous growth, and mixed culture involves two or more species living together. The straight plate method is one of a few methods that is used to isolate bacterial species and allow for single cells to be obtained so individual colonies can form. And the term inoculation refers to introducing a microorganism into a sterile medium. Now, into culture medias, selective medias inhibit growth of certain species, specifically gram-positive bacteria. And in the case of MacConkey agars, gram-negative bacteria primarily grows in this situation, while in differential medias, there's substance that can be contained that microbes change in identifiable ways. So you have MacConkey agars in this case that bases gram-negative bacteria on the ability to ferment lactose. Pink or red means that the microbe can ferment lactose, while colorless, there's no fermentation of lactose. For antimicrobials exhibit a bacteriostatic effect, which means that it halts or inhibits microbial growth, while for bactericidal effects, that kills off bacterial growth completely. And a good example of this is Penicillium rubens, which is a kind of mold, and this is usually used for antibiotics such as penicillin. The zone of inhibition is an area in which no growth occurs for the organism, and the size of this zone is impacted by the concentration of the antibiotic and the solubility and the diffusion rate.
All right, next we'll be talking about the cell environments in lab. So cell cultures, they must be kept under certain conditions for optimal growth, and depending on the cell that you're working with, the conditions will vary. So the environments are kind of separated into two different sections. So we have like the physiochemical environment and the physiological environment. So the physiochemical environment involves growth media like pH, osmotic pressure, and oxygen and carbon dioxide levels, and as well as the temperature as well. And the physiological environment includes hormone and nutrient concentrations. And the physiological environment isn't going to be as well defined as the physiochemical environment because we're able to be more defined with the things that we're able to alter. And to delve a little bit more specifically into these factors, so for pH levels, most mammalian cells are able to grow best at a pH of around 7.4. And there's not a lot of variability with this, so it won't stray too much from that number. And insect cell lines grow best at a pH of around 6.2. As far as carbon dioxide levels, most researchers use 5 to 7% carbon dioxide, but 4 to 10% is the most common for cell cultures. And it's also important to note that changes in the atmospheric carbon dioxide can actually alter the pH of a medium, which makes it necessary to use oxygenous carbon dioxide when you're using a media buffered carbon dioxide bicarbonate based buffer. And for temperature, most human and mammalia cells are maintained at 36 to 37 degrees temperature for the best growth. Insect cells are cultured at around 27 to 30 degrees Celsius, while cells that come from cold-blooded animals are able to tolerate a more wide range of temperatures, like 15 degrees to 26 degrees Celsius. And researchers are able to keep these temperatures accurate and consistent using incubators. Let's use that. And then for our Bible verse, we decided to go with Genesis chapter 1, verses 20 and 21, which says, "And God said, let the water teem with living creatures, and let birds fly above the earth across the vault of the sky. So God created the great creatures of the sea and everything living with which the water teems and that moves about in it, according to their kinds, and every winged bird according to its kind." So we went with this one just because we felt like it reflected like the diversity of the world that God created. And obviously this kind of relates back to microbiology with all the different microorganisms and whatnot. So yeah, and then these are our references.